The tissue engineering in periodontology was proposed by Langer and Vancati in 1993. As defined by Langer and Vancati, tissue engineering is an interdisciplinary field that applies the principles of engineering and life sciences towards development of biological substitutes that restore and maintain or improve tissue function or a whole organ. So the two approaches to tissue engineering, one is an ex vivo approach and one is the in vivo approach. In the ex vivo approach, the cells are cultured in the laboratory where you need a biodegradable scaffold. In the presence of growth factors, the cells are developed. So for this kind of regeneration, you need the availability of the cells, and second one is the presence of signaling molecules. Next is your in vivo approach. In this technique, all the components which are required for regeneration are placed in the tissue defect, and an environment which is conducive to maximum regeneration is created to achieve favorable regeneration. The tissue engineering approach to bone and periodontal regeneration consists of three key elements. One is stem cells, scaffolds, and signaling molecules. The cells can be adult stem cells, embryonic stem cells, periodontal ligament derived stem cells, or dental pulp stem cells, or marrow stroma stem cells. The scaffolds can be collagen, fibronectin, fibrin, hyaluronic acid, proteoglycan, foams, fibers, gels, and membranes. And the signaling molecules can be your transforming growth factor, your platelet derived growth factor, your fibroblast growth factor, etc. The criteria for successful tissue engineering is a biomechanical property and biological functions. The scaffolds, the architectural geometry, the space maintaining properties. In biological function, the cell should be recruited and should be able to proliferate. It should be able to survive in the culture at the site of neovascularization. So cells. Going on to cells, we have stem cells. Stem cells are immature progenitor cells capable of self-renewal and multilineage differentiation through a process of asymmetric mitosis that leads to two daughter cells and one identical to stem cells, daughter stem cells, and one capable of differentiation into more mature cells, progenitor cells. Stem cells are divided into embryonic stem cells and adult stem cells. The embryonic stem cells are divided into derived from the blastocyst and totipotent cells. The adult stem cells are multipotent cells and they are hemopoietic stem cells. The stem cells can be totipotent, pluripotent, or multipotent. So this is the diagrammatic representation of your stem cells. Dental pulp stem cells are present, shed is present, then also you have periodontal ligament stem cells, scap is present, dental follicle stem cells are present. Scaffolds provides a three-dimensional substratum on which the cells can proliferate and migrate, produce a matrix and form a functional tissue with a desired shape. Naturally derived synthetic scaffolds are ceramics, hydroxyapatite, tricalcium phosphate, natural polymers like hyaluronic acid, alginate, agarose, collagen, albumin, and chitosan. Synthetic are polymers, synthetic polyesters, polyglycolic acid, polylactic acid, polycaprolactone, copolymers of polyethylene oxide, polypropylene oxide, PLGA as a copolymer of PGA and PLA, and PLGA copolymer foam. Signaling molecules are proteins that may act locally or systemically to affect the growth and functions of the cells in various manners. The two types of signaling molecules are growth factors and morphogens that act by altering the cell phenotype, causing the differentiation of stem cells into bone-forming cells, a process commonly known as osteoinduction. Growth factors act on external cell membrane receptors of target cells, provide the signal to local mesenchymal and epithelial cells to migrate, divide, and increase the matrix synthesis. The growth factors have received the most attention in hard and soft tissues. Bone morphogenic proteins are the members of transforming growth factor superfamily. They possess the unique ability to induce cartilage and bone formation. Bone morphogenic proteins was given in 1965 by Urist and colleagues to the active components in demineralizing bone and bone extracts which are capable of inducing bone formation at the ectopic sites. Reddy and Huggins in 1972 found that bone extracts induce bone formation at the ectopic sites. In 1988, the first BMPs were isolated. It was cloned by Wani et al. At least 7 BMPs have been isolated. In the field of periodontal regeneration, BMP 2 and 7 plays an important role. So you have the BMPs are dimeric molecules, that is a single intermolecular disulfide bond for biologic activity. The monomeric subunit has about 120 amino acids and 7 cysteine residues. It is divided into 4 subfamilies. One is BMP, 2 and 4 is comprises of group 1. In group 2 you have BMP 3 and 3B. In group 3 you have BMP 5, 6, 7 and 8 and others in group 4. Functions of BMP. It regulates various mesenchymal and osteoblastic-like activities, chemotaxis, anchorage dependent cell attachment, cell replication, differentiation of osteoblast, alkaline phosphatase activity and osteocalcin synthesis or mineralization. BMP also plays a critical role in tooth morphogenesis. 2, 4 and 7 are expressed in the dental epithelium. Recombinant BMP 2 and 4 can be used as a substitute for dental epithelium in inducing mesenchyme differentiation. BMP 3 and 7 have been immune localized to developing periodontal ligament and cementum. Other BMPs, BMP 2 was localized only in the alveolar bone. The bone morphogenic protein carriers. One of the major obstacles to the clinical use of BMP is the challenge to define the optimal delivery system. A BMP is soluble in extracellular solution. It must have a carrier without which it is phagocytosed within 10 days. So although a matrix carrier is not essential to promote bone formation, there are a number of advantages to an appropriate carrier, including localization retention of BMP to the site, providing a three-dimensional extracellular matrix scaffold for mesenchymal cell differentiation, a shape that may help and define the resulting new bone, and providing a substrate for cell growth and differentiation. The carrier material can be in the form of blocks, granules, paste, solutions, or a self-setting cement. Receptors for BMP. A comparison of other growth factors reveals BMPs are 34 to 38% related to the transforming growth factor beta family. BMPs and transforming growth factor beta have cognate BMP type 1 and type 2 receptors and TGF beta type 1 and type 2 receptors respectively. The BMP type 1 receptor protein kinase phosphorylates intercellular signaling substances SMADs, a fusion of SMA gene in a Cenorvidus elegans MAD gene. The transforming growth factor beta 1 receptor kinase specifically phosphorylates SMAD2 and 3. The phosphorylated 1, 3 and 8 BMP signaling SMADs 2 and 3 transforming growth factor beta signaling SMADs partner with the SMAD4 to form signaling complexes in the cytosol that enter the cell. Although BMPs and transforming growth factor beta ligands signal via distinct receptors, they act in collaboration during bone and tooth morphogenesis. The production of recombinant human bone morphogenic proteins. Recombinant proteins are produced from one of the several cellular expression systems of the bacteria, insect cells and mammalian cells. RhBMP2 is produced using a mammalian cell expression. BMP in periodontal regeneration. In the field of periodontal regeneration, much of research interest has focused on BMP 2, 3 and 7. The first human study using a BMP to promote periodontal regeneration utilized a single application of BMP 3 osteogenin combined with a demineralized bone allograft in a submerged tooth model. So there are various authors with various concepts. Healing through ankylosis has been a concern, so most of the recent research utilizes recombinant human BMPs as involved in the preparation of implant site for osseointegration. Then BMP 7 plays an important role in dental implant wound healing. And in the recent study, combined adenovirus mediated human BMP2 gene modified bone marrow stromal cells with stem cells with allograft enhance the defect healing and improve the strength of implant fixation with osseointegration. Bovine BMP tested in dog model has shown to increase the rate of osseointegration around cylindrical uncoated or endosseous implants. The tissue reactions to titanium implants coated with BMP was further assessed by scanning electron microscopy for 12 weeks in the same dog model. The results reveal abundant lamellar bone formation around the BMP coated implants. Potential challenges yet to overcome. Notwithstanding these significant advances, there are still numerous biological, technical and clinical hurdles to overcome. Complete understanding about the molecular and signaling pathways that form the ground for tissue engineering and underlying cell renewal and differentiation is yet to obtain. As cell culture medium often requires xenogenic products, cell cultures may not be completely free of pathogens. Culture conditions are not sufficiently developed to mimic the cell microenvironment, and cell-based periodontal therapies lead to immune rejection after administration of cells into the host. A potential solution to this problem lies in the use of autologous stem cells to overcome the immune rejection. So, going to conclude, several preclinical studies have shown that RHBMP2 induces normal physiological bone in clinically relevant defects in craniofacial skeleton. Recombinant protein morphogens recently provided by FDA such as BMP2 and BMP7 could be applied in dentistry to facilitate the repair of craniofacial structures. So further studies are needed for the development of carrier material that have mechanical properties and surgical practicality appropriate for controlled release of BMP. We need to look beyond before we can achieve the dream. Tissue engineering has enlarged our vision and thus made the fascination of being able to achieve regeneration of periodontal complex in its entirety a reality. Though the task has been arduous, but promise still remains. Thank you for the patient hearing.